So you may recall from the previous video that inside of the periosteum we have osteoblasts and osteoclasts. Let's talk about what those cells actually are. So in this, the first cell that you're going to be coming across is an osteogenic cell. I wouldn't worry about this one too much. It is the stem cell which differentiates into these guys. So it's kind of like the precursor cell, if you will. The first cell we're going to be worrying about is called the osteoblast. The second one is the osteoclast, right over here. We've already studied the osteocytes in lab. And remember that I said it has kind of a central cell body, or not a cell body, but the central part of the cell rests inside of a lacuna or a tiny lake, and then it sends out little tendrils, and its job is to maintain the surrounding bone tissue. So that's called the mature bone cell, the osteocyte. But now the osteoblast it's usually nicknamed as the bone builder. It basically secretes a substance called osteoid, which we will discuss next week. Osteoid is secreted by the osteoblast, and it actually creates bone matrix. And then the osteoblast becomes an osteocyte. It basically buries itself inside of the osteoid and becomes an osteocyte. But until it gets buried, it's called an osteoblast, and it continues to create bone material. So it's a bone builder. The osteoclast has the opposite role. As you can see, it's called the bone resorbing cell. We usually call this the bone destroyer or the bone eater. And the osteoclast breaks down bone material, which you'd think, well, that's very damaging. And it can be, and it can actually be very painful. But it's a very important part of the way that bone works. So keep in mind, down the line, we're going to be talking about bone remodeling. Both purposes are very important. You need the osteoblast for bone building, but you also need the osteoclast to remove bone material that is not needed in that region. Both of these are very important for that. So remember, osteoblasts, bone builders, osteoclasts, bone destroyers. And all three of these are living cells. Osteoblasts, osteocytes, and osteoclasts are all living cells. The osteoblasts and osteoclasts tend to live inside of this guy here, the periosteum. So that's where you're going to find the most bone matrix being added on or taken off, which makes sense. It's happening sort of at the outside of the bone. So I would suggest that there will be one question on the test that will talk about projections and depressions and openings. And these are all called bone markings. So table 6.1 is going to show you bone markings. You don't have to get to know each and every one of them, but basically I want you to know one or the other. Like you want to know an example of one, and you want to know an example of the other one. Now projections are things that kind of pooch out and stick out of bones. And these are not accidental. There's a reason why they're all there. As strange as they look, they're usually attachment points or projections that help two bones actually fit together better, or places where muscles attach and they've created stress and they've actually created a tug on the bone and it's actually started to grow out in that region. So examples of these, you can take a look at the line. Ones that we'll discuss here are the trochanters. These are attachment points for muscles, for example, or the heads that help to fit into other bones. This is actually this one right here is the head of the femur, and it fits into this little guy here, which is called the acetabulum of the pelvic bone. So this is a projection. This guy is a depression. So in the next, so we have a few more projections here, and then there's also some depressions and openings. Depressions and openings will be places where, for example, two bones will fit better together by creating a depression or a bowl. Nerves and bloods may travel through and around a bone through literal holes in the bones. And those are some main reasons why you would have depressions or openings. So go ahead and take a look at this list as well. Remember that we have, I think this is a pretty good picture to see this particular aspect. We're going to be looking at the microscopic look of compact bone. And so in this picture we have both spongy bone and compact bone. Remember that both will be present in all bones. Now the compact bone is going to have these structures that are called osteons. And I like this picture because it actually shows them in relief in here. It shows them how they actually look inside of the bone itself, which is pretty cool. This is what it actually looks like. If you were to pull one of those osteons out of the bone, you'd actually see those layers that surround the central canal. This is a nice picture of them all stacked together, and one that is pulled out, so you kind of get the idea here. An osteon is the structural unit of compact bone specifically. There's a series of lamellae that wrap around each other. Each of these are made of collagen fibers, and they're made of alternating directionality of collagen fibers. So not only is it strong because it's mostly made of collagen, but the lamellae themselves are alternating directions so that it really resists twisting force. It's very, very hard to break a bone. It is possible, but it's very difficult to do because they are incredibly strong and because of the directionality of those lamellae. In this picture, of course, it's quite obvious to see the Haversian canal, or the central canal. Here you can see the blood vessels and nerves making their way through there. And again, this is a pretty good picture that shows, again, an osteon here pulled out, the Haversian center, or the Haversian canal or the central canal in the center. And close up, you can see the nerve, vein, and artery. Very vascular tissue here. You can see that every single one of those Haversian canals has blood vessels and nerves. So very vascular. You'll see those canaliculi here. This is probably the best picture that's showing the canaliculi. You'll see osteocytes sitting inside of the lacunae, those tiny lakes in there. You'll see in this sketch, you'll actually be able to see the little living cells inside of there, and you'll be able to see the way that they send out their tendrils into the surrounding matrix through the canaliculi, the tiny canals. So the canaliculi will connect the lacunae eventually through a series of canaliculi to the Haversian canal in the center. And again, this is showing those osteocytes like in isolated, so without the bone material, you can see how they stick out their little arms through the canaliculi. The last structure we want to take a look at are the perforating or Volkmann's canals. The fact is that, although it's not shown in this picture, you have to get blood vessels and nerves into the tissue in the first place. So here we're showing that the blood vessels and the nerves come from the periosteum on the very outside of there. But, you know, they have to continue on in a horizontal manner. So there are canals in here called perforating canals that run horizontal to the length of the Haversian canals, or the central canal. So here's a central canal, and here's a perforating canal that helps to connect all of these structures with each other.